This is an overview of an article entitled Bivariate Genome-Wide Association Scan Identifies 6 Novel Loci Associated with Lipid Levels in Coronary Artery Disease, published in the December 2018 issue of Circulation, Genomic and Precision Medicine. Genome-Wide Association Studies, or GWAS, for coronary artery disease, or CAD, have identified numerous susceptibility loci associated with the disease. These loci account for only a fraction of the total heritability for CAD, indicating that additional loci remain to be discovered. In addition, many CAD-associated regions lack an obvious mechanistic connection to heart disease. A recent trend to identify novel mechanistic connections is to utilize data integration, whereby associations for a phenotypic endpoint such as CAD are statistically analyzed jointly with additional data types to better understand the underlying phenotypic association. In addition, large-scale studies have now emerged which map genetic variation associated with changes in gene expression, that is, expression quantitative trait loci or EQTLs, across major human organs and tissues. These data provide an opportunity to link a CAD association signal to a putative causal gene and potentially a tissue of action. Multivariate studies that combine association data from multiple traits can help interpret the underlying associations at existing loci and have higher power for novel locus discovery than univariate studies. The authors of the paper under discussion performed a bivariate GWAS scan to discover novel loci associated with CAD in any of 4 lipid-related traits: LDL cholesterol, HDL cholesterol, total cholesterol, and triglyceride levels, using CAD association data from the Cardiogram plus C4D Consortium and UK Biobank, and lipid association data from the Global Lipids Genetics Consortium. To focus on a set of loci that potentially point to a shared causal variant and cognate gene of interest across traits, they applied statistical co-localization for the lipid and CAD association signals, both with each other and with gene expression datasets. In this way, the authors identified variants in 6 novel genome-wide significant loci. The first variant, RS2908290, was associated with triglycerides, or TG, and CAD, and is in an intron of the GCK, or glucokinase gene. Prior evidence supports GCK as the causal gene at this locus, as well as associations with glycated hemoglobin and type 2 diabetes mellitus. The second variant, RS895953, is in an intron of the SET1B gene. It had bivariate associations with TG and CAD, as well as HDL cholesterol and CAD. It is an EQTL for multiple genes, including HPD and RHOF, as shown here for adipose. The third variant, RS3741782, was associated with LDL cholesterol and CAD, and falls within an intron of the CORO1C gene. It is an EQTL for CORO1C in visceral adipose tissue and in the adrenal gland, and for SSH1 in whole blood. Both genes regulate actin reorganization. The fourth variant, RS124236664, was associated with TG and CAD and is located in an intron of FBRSL1. It is an EQTL for this gene in both pancreas and whole blood. The fifth variant, RS1378942, had bivariate associations with CAD and total cholesterol, and to a weaker degree with LDL cholesterol, along with an association with systolic blood pressure. It is an EQTL for multiple genes in multiple tissues. A sixth variant, RS703334, not shown here, was associated with LDL cholesterol and CAD without an obvious link to an EQTL. In conclusion, the authors identified 6 previously unreported loci with bivariate genome-wide significant associations with 1 or more lipid traits and CAD. At several loci, GWAS signals jointly localized with EQTLs for 1 or more nearby genes, indicating effects on disease risk through regulatory activity. Further functional experimental work will be required to verify and elucidate these candidates' associations with lipid and CAD pathways. Nonetheless, the authors demonstrated the power of data integrative approaches to discover novel loci, influencing phenotypes of interest.